It may or may not be obvious, but if we want to connect monosaccharide subunits, so these individual smallest unit of carbohydrates, we need to do so through some kind of a chemical bond. And so if we look at these two monosaccharide subunits, here we have two glucose molecules. And highlighted are the OH groups that are going to participate in this bond, which is a glycosidic linkage. And when we do this, we leave one oxygen that's going to connect and make up the glycosidic linkage. We lose two hydrogens in the other oxygen, and we form water. And now we have a disaccharide. This is a linked molecule. We have a compound sugar, a 2-unit sugar, and we've completed a dehydration reaction, right? We saw this previously when we are looking at how biological molecules are joined together. Very often a water forms in this type of dehydration reaction. It's very convenient, obviously, because, water is all over our body, so creating this as a byproduct is no harm at all. But key here is the glycosidic linkage. We have two monomers that come together to form this dimer. Okay, and when we have our monosaccharides, again, the simple sugars, glucose, fructose, galactose, this is great. Disaccharides, these are double sugars, maltose, sucrose, or lactose, constructed of two of these individual simple sugar units and have formed a bond. And we'll look a little bit more at this glycosidic linkage, what it means, and what impact it has on the resulting carbohydrate coming up here, right? And note that here is two different molecules of glucose, right? First, we have alpha glucose, and second, we have beta glucose. And how do these differ? Well, we have everything in the same position, except when we look at alpha glucose, this OH on the starred carbon, which is the important and active carbon for adding on another monomer unit, this is pointed down. And in beta glucose, this OH is pointed up. And this is going to impact how the resulting bond is going to be formed from these two different structures. They may look very similar, but these are actually two different molecules. These are structurally different, even though all the bonds are still connected to all the same molecules. And the result is this, is that when we have the first version with the OH group pointing down on the glucose, we end up getting a 1,4-alpha glycosidic linkage, we'll call that. And this is where the bond points down for both of the two individual units. As you can see in this depiction, the first carbohydrate unit has the highlighted downward bond from its 1 carbon. That's the number that carbon gets. And then if we go around the ring, we get to 4. That's where the second link is formed. So that's why it's called 1,4s. It's from the 1 carbon of the first monomer unit to the 4 carbon of the second monomer unit. And it's alpha because both of the bonds are pointed down. This gives a special structure to this linkage. Alternatively, if we have the beta glucose that's used where its 1 carbon OH bond is pointing up, we're going to get a beta 1,4 glycosidic linkage where we now have the bond going up from the 1 carbon of the first monomer unit and down from the 4 carbon of the second monomer unit. And so this makes a different dimer, a different 2-joined carbohydrate, a different 2-unit carbohydrate, right? It's all the same stuff, but the way that the bonds are constructed have huge implications and is a really important thing to know when we're looking at carbohydrates. These continue to grow. We get polysaccharides. This process grows into prevalent polysaccharides like cellulose, starch, glycogen. We've talked about starch and glycogen already. These are for storage, and cellulose is a little bit different. And so when we have the glycosidic linkage, this is going to affect both the structure and the function, right? Cellulose we'll look at very soon. Sorry to skip over that one quickly. But the way we connect these is going to make different structures. And so let's look at the following examples. First is cellulose, right? We mentioned cellulose. In cellulose, we have alternating linkages where we have kind of up and down, and this isn't sort of a nice, smooth, all 1,4 linkage structure. We have, or 1,4 alpha linkage structure. We have some variations here. And because of these variations, cellulose makes these long, thin fibers where it ends up being used for things like the cell wall and stuff that's harder to break down. Not all animals can break down cellulose, which is to say, like, not all animals can eat grass. But amylose, this is used for storage in animals. This is all those nice 1,4 glycosidic linkages. And this is what what plants use for, more easy digestible food. And this is, that kind of easy starch that when animals consume a plant product, they're kind of looking for something more like amylose. The way the glycosidic linkages are formed affects these structures. Amylose makes a coil compared to the fibers. That makes it easier to break down. Okay, classification of carbohydrates. Take a look at this, right? Monosaccharides, disaccharides, polysaccharides. Mono, di, poly, single sugar, 2 monosaccharides, many monosaccharides, and the examples that are on this we've looked at previously. Monosaccharides are the simple sugars. That's what needs to be absorbed by the body. We'll talk about that soon. So if you're digesting something that's a disaccharide or a polysaccharide source, breaking that glycosidic linkage, as we'll see in the next part, will be essential before you can unlock the energy of the monosaccharide.